So today we're for our February external speaker to the seminar series. Our distinguished speaker is Dr. Kirk Knowlton, and this is sort of a personal, real pleasure for me to introduce Kirk, because not only am I very close to Kirk, but I feel embarrassed to say that because he really has mentored me for very many years. And so I feel like I'm introducing one of my mentors. Certainly, Kirk is one of the big reasons I came to the University of Utah, without question. So if you start having sort of tomatoes and fruit thrown at you, you'll understand why. But I first met Dr. Knowlton when he was chief of cardiology down at University of California, San Diego, and grew what remains today one of the best academic cardiology divisions in the country. Frankly, if not the, when one focuses on science. The people it's produced, the people are there now, whether one quantifies it by just the personalities involved, the funding they've generated, the papers they've generated is just remarkable. And then you came to Intermountain in 2016 already. All right. Personally, and the other way I've gotten to know Dr. Knowlton is through his research, where he's done a lot of work on viral myocarditis, myocarditis in general, which is now, obviously crossed over to both COVID and what we now know as immune system and sort of cardiac immunology, if you will. And that's a field that I don't think I would have understood those terms only 2 or 3 years ago. And certainly the COVID pandemic has made us all very, very aware of how important immune regulation of the heart is. That's reason enough to be very honored to have Kirk join us. One of the other reasons, to be very transparent, is that as a collective community here, we're building the Salt Lake City area, and we're building the University of Utah, Intermountain, and the state of Utah. Okay, and we should all work towards working more closely together. When I was at UCSF long ago, there was always that creative dynamic between UCSF and Stanford. Which they've always been enemy, and that's never changed. Although at some point they were formal, they were on the same team. Didn't last long, but but the point is the faculty and the researchers have always maintained close relationships, and even the administrations have maintained close relationships. And if we could, we need to do even more of that. With Intermountain. That certainly happens on the clinical side, and we need to build it up on the research side. And so we each have our strengths, but the more we work together, the more we're going to build the community, the more we're going to build the state of Utah, and that's what we're about. Okay, and certainly we're about advancing cardiovascular disease. Currently, Dr. Knowlton, I can spend the whole time reading your titles, is chair of cardiovascular department, as well as the associate medical director of cardiology, medical director of cardiovascular research, professor of clinical investigation, Intermountain Healthcare, and has an appointment here in the Department of Internal Medicine as well. So the title of the talk is Myocarditis from Coxsackivirus to Cytoskeleton to Coronavirus. Those of you who know me will understand that most of those terms are near and dear to my heart. And so it's a big honor, truly a big thrill to have Dr. Knowlton join us today, and looking forward to your talk. Thank you.
Thank you, Robin. I feel a little bit like I'm coming home when I come to visit here, having grown up in Utah, as many of you know, and gone to the University of Utah for medical school, but mostly because there are friends here, and that's what counts in the long run. And certainly Robin, as he mentioned, has been one of my close friends through the years. And you know, I'll never forget what a positive, happy day it was when he called me and he said, "Kirk, you'll never believe it, I'm thinking of coming to Utah." And I said, "Oh my goodness, that's outstanding. Anything I can do to help." And so it's so wonderful to have him here and to be able to have this friendship and this interaction. And he and I both share the vision that our mission here really is for the people of the state of Utah. How can we best care for the people of the state of Utah, either through our clinical research, our basic research, our translational research, or even better, a combination of all of those things? And how can we make it so that the people here and the care and the research done in the state of Utah is as good as or better than anywhere in the United States? And that's really a wonderful opportunity. So I'm grateful for that. I'm grateful to be here for all of those reasons. You know, my move to Intermountain probably deserves a brief moment as to why would one move from having an NIH R01 funded laboratory with a training grant and at one point a PPG and chief of cardiology to Intermountain Healthcare. Well, I have to admit, yes, having the fact that it is in the state of Utah and my parents were getting older made a difference. But I was really looking forward to being able to do clinically related research. And that's been really an asset. We have many samples, about over 35,000 DNA and plasma samples out of over 22,000 individuals we've now sequenced in collaboration with Decode and Heredigene, 22,000 individuals. We have whole genome sequence on about 10,000 of those. And we're just starting to publish papers now. We just were included on a paper on the genetic causes of vertigo, interestingly enough, from the samples that we contributed there. So it has been a good move. But really what I'd like to talk about more than anything is some of the work that some of you may be interested in. It's a little bit of a story that begins a while ago and plays through, but I think it's playing out. And I think that the hypotheses and some of the theories we have are developing and growing even further. So let me tell you a bit about that, if I may. Let's see if I can figure out how to advance here. Okay, so fundamentally, my goal is to try to help us think differently about myocarditis, which probably is not exactly the right word for the disease process that we're thinking about, because it limits our thinking a little bit too much. But I think that that is a first piece. I'd like to talk a little bit about genetics and myocarditis and lessons that we've learned from mice and now humans. A little bit about immunomodulation of myocarditis and pericarditis, transition to lessons learned from COVID-19 pandemic and COVID-19 myocarditis. And is it myocarditis or not myocarditis, which is a controversial subject, actually. So first of all, if you want a general review of myocarditis, I point you to this paper in Nature Reviews. I won't touch entirely on myocarditis. This is not a clinical talk. I'm going to talk about things that related to myocarditis that I'm interested in. So I think, as I mentioned, I think it's time to think a little bit differently about myocarditis. We need to expand our brains in that regard and our investigation. And I'll give you some thoughts as to how what I'm thinking. This is the traditional. Sorry, let's go the other direction. The traditional picture of the men with the blind men and the elephant, of course, I think applies to this field too, because people have different viewpoints of what myocarditis is, depending on whether they're feeling the tail or the tusk or the nose, the trunk of the elephant. So is it virus? Is it cytokines? Is it heart failure? Is it T cells? Is it autoimmunity? Cardiac magnetic resonance imaging is the big thing. Dilated cardiomyopathy. What about Dallas criteria? Arrhythmias. All these are pieces of myocarditis in reality. So the traditional view of myocarditis is that a person gets infected with a virus. That's the most traditional view, going back to the late 60s and 70s when we learned we could infect mice with a virus and cause myocarditis. And we then began to be able to detect virus in the heart in myocarditis that was actually back in the 50s when that was done. But basically the virus can infect a person and would then go to the heart in many cases and infect the heart, causing myocardial damage to the heart. With that myocardial damage, the damage itself may be sufficient to cause these, but it usually will also result in viral mediated immune activation, in which there are lymphocytes that infiltrate into the myocardium and cause dysfunction. It's also possible that the person has activation of an autoimmune syndrome that could also bring in lymphocytes, all of which have a negative effect on the heart. Okay. You're not able to see my mouse, I guess, right? So how about if I hide the presenter view? Let me see if
do a cursor. Yeah, there, it's pointer.
No.
You was there. I just saw it for a second.
Did you see it there?
Or you can change it.
Yep, right here.
Okay. They see it there, but not there. All right. So I may have just kind of messed things up. I can't see anything here now, or the slides I have
Yeah,
here.
you don't say
Okay, so let me tell you about a patient that helped me to develop a sense that why I wanted to study this. This was a 35-year-old man who came into the hospital with moderate to severe dyspnea, preceded by an upper respiratory infection. He was treated several weeks for a pneumonia. They didn't realize that he had heart disease or heart failure. Finally, a diagnosis of congestive heart failure was made with severe left ventricular systolic dysfunction. He was actually sent to UCSD for an LVAD implantation. Myocardial biopsy was performed given the severity of the illness, and 3 samples went to the laboratory for histology. The official read was no evidence of myocarditis. A 4th sample came to my laboratory, and it showed the following. Here you can see an obvious example of myocarditis. There's no question that this person had myocarditis, as shown on this biopsy, with infiltration of the heart muscle, with lymphocytic infiltration, myocyte damage. It met all the criteria needed to make the diagnosis of myocarditis, and he ended up not needing a left ventricular assist device. His immunostaining was positive for Coxsackie virus. So this was classic model Coxsackie virus myocarditis. So at that point, what we knew and didn't know, we knew that he had met Dallas criteria for myocarditis, probably caused by Coxsackievirus, but we don't treat him any differently than any other. Will immunotherapy help? At that point, probably not, thanks to studies led here at the University of Utah, the myocarditis treatment trial that showed no real benefit of that. And why did he get the myocarditis in the first place? We didn't know that. Why he would get it and other people with Coxsackie might not. And is there actually a genetic predisposition here? So this took me to some of the experiments that we did that related to the cytoskeleton and their interaction with Coxsackie. The reason we looked at dystrophin is a couple-fold. One is dystrophin deficiency is known to cause Duchenne muscular dystrophy. There's very well characterized cardiomyopathy that occurs in those young people. They often die of heart failure because of the dystrophin deficiency. So we started to wonder whether the virus could interact with cytoskeletal proteins like dystrophin. And we found a model, a computer-generated model that predicted that one of the viral proteases would cleave the dystrophin molecule. So we set out to address that question. Could the enterovirus infection affect the dystrophin glycoprotein complex? And ultimately, we came to this model here, which was that shown on the top is the fact that this is the Coxsackie polyprotein that is translated in a monocystronic polyprotein and then cleaved into its individual parts in cysts by protease 2A. In this portion right here, also protease 3C cleaves some of the other portions. There was a very specific cleavage site for protease 2A in the dystrophin molecule. And we demonstrated that that was the case and proved exactly where it cleaved, et cetera, a body of several papers that we published on this, including this one here, which was published in Nature Medicine, that showed that, that in cells, blue, that were infected with Coxsackivirus, there was an absence or disruption of the dystrophin glycoprotein complex in those cells that were infected, as you can clearly see there. And the red demonstrates an Evans blue dye permeability of the in these cells that were infected where the dystrophin was missing. So they're infected, there's disruption of the dystrophin glycoprotein complex, and they are more permeable. So we then specifically asked, could there be an interaction between genetic and acquired causes of cardiomyopathy? Can the acquired factors worsen the genetic causes of disease? So the genetic disease in humans is dystrophin deficiency. The mouse model for that is MDX mice. And so we tried to infect those mice. This is the key paper on that. It was also published in Nature Medicine that showed that in fact, this is the dystrophin competent mouse on the left, and on the right is the dystrophin deficient mouse heart. And the white in this case is Evans blue dye, just taking a picture in black and white because it showed it more clearly. You see that there's much more Evans blue dye, and we could quantify that as well, shown on the bar graphs. And also there was a higher level of troponin release into the serum of the patients that were dystrophin deficient. That demonstrated that in fact virus infection had a much worse prognosis, or had much more severe pathogenesis when the dystrophin molecule was absent. So that started to change our thinking to the virus doesn't just infect a person, it affects a person with a genetic predisposition potentially to virus infection. We don't know what all those predispositions are, but that could be a factor in that process. So the dystrophin deficient data demonstrated that in dystrophin competent cells, they have a stable membrane thanks to the dystrophin complex that's present there. And when they get infected, there's viral replication. Dystrophin deficient cells have a fragile membrane because that dystrophin molecule is absent, and they get infected, also have viral replication. But in this case, it requires cleavage on the dystrophin competent cells, it requires cleavage of dystrophin and leading to increased membrane permeability. But on the dystrophin deficient on the right, there's very efficient disruption of the cell membrane because there's no real need for cleavage of dystrophin. And so essentially all the virus exits the cell in the dystrophin deficient and increases viral replication, which we demonstrated in other experiments that I'll not show you today. So to prove this really was the factor and not just kind of an association, we took, one of my postdocs took on an ambitious project that took many, many years to do a knock-in. At that point in time, it took many years, probably much easier now, to do a knock-in at the dystrophin cleavage site of protease 2A. So we knocked in to make it so it was not cleavable. And that was in exon 50 is what's shown here. And these are the proteins that or the amino acids that we generated as a result of that, leading to a mouse where the only difference between wild type and the knock-in was that the one could not be cleaved by protease 2A. The mutant mice could not be cleaved by protease 2A. And so we see here that was the case. This is a wild type. This is demonstrated dystrophin cleavage that could not take place with the in the dystrophin knock-in mice and so on, and that there was infection there in those mice as well. And here's another picture of that in actual mice, not just the cells, where the wild type mice, The EIF4G1 could still be cleaved, but dystrophin itself could not be cleaved. And there was, my battery's running low. Do we want to plug this in? Other proteins were still cleaved as expected. And there was evidence of infection in those mice, even though it was not cleaved. So going forward here, and there again is the disruption of the cleavage. So in these mice, basically what it demonstrated is that when dystrophin cannot be cleaved, dystrophin knock-in, Ki, as shown there, that doesn't transmit online, does it?
No, this is on present.
That's okay. Knock-in, there are many fewer Evans blue dye positive cells, i.e. many fewer that have disrupted cell membrane, whereas in the wild type is a lot of Evans blue dye positivity, and that's shown there in the red staining in the 4 panels that you see there. So clearly, if we only remove the cleavage of dystrophin, we get less disruption of the cell membrane, and that's shown in this other marker of cell membrane permeability known as the von Cossa stain. If you're looking at cell membrane permeability, I highly recommend this one. It's a very nice stain, black and white. So we saw that there as well. And there was decrease in virus titer as highlighted there on the 8-day heart, whereas the other organs had normal levels at day 4, but as is usually the case in the other organs, it decreases dramatically by day 8. So it affected viral replication as well. And there was less myocarditis, although there was a little bit more than I would have thought, but there was less. So this is the model that we developed, that in a normal mouse, shown on the panel on the left, protease 2A cleaves dystrophin and then disrupts the cell membrane, lets virus out of the cell, and so it can infect other cells. When we block the cleavage, as shown on the right, with knock-in, then the virus has difficulty getting out of the cell, and it cannot infect other cells to continue the infection. But we'll come back to why that may be important, not only in MDX, but in other clinical models as well. And this is one of the first examples that maybe this is important. This was a paper published several years ago now that showed 20 Duchenne muscular dystrophy patients who had a higher incidence of myocarditis based on CMR. And those with myocarditis had rapid deterioration of the left ventricular function, CMR being cardiac magnetic resonance, as you probably know. Rapid deterioration of left ventricular function during the next year. So there was evidence of Duchenne myocarditis in those people, those kids who had Duchenne muscular dystrophy. Why they suddenly deteriorate is not known. And this is an example of some of the myocarditis on cardiac MR and histology with those kids. So now our view again focuses here on the person with the genetic defect or abnormality. And I think that additional evidence clinically came from this paper. There have been one or two others since that are similar, that demonstrate autosomal recessive cardiomyopathies can present as acute myocarditis. And this is a pediatric population that had acute myocarditis. There's an increase in the percentage who have homozygous or compound heterozygous variants in genes that have been associated with dilated cardiomyopathy. These are people that present with myocarditis, but they also have a higher rate of some abnormalities, including desmoplakin, plakophilin, troponin I type 3, and others that you see there as well. The code for cytoskeletal contractile proteins. You're stuck. I'm stuck. Let's see what we can do here. That's not
Okay.
not going forward. I shouldn't have taken it out of his interview.
Okay. There you go. How did you advance it? Okay, let me.
Missed all my great data.
We believe it. Can I just close it and?
Sure, if you'd like.
Yeah, because it's.
Let's just leave it there. Yeah. Let's see if I can talk it into giving me pointer here. Pointer. Okay.
There you go.
Let's march quickly here if we can.
Okay.
Did you see my knock-in data? That was really
Sort of
good.
knock-in. We sort of did that.
That was a culmination. That was a culmination of about 8 years of work.
Here's
So-
where we got frozen.
Here we got frozen.
We
Okay.
know protease is bad.
Very good. And
Leave
cleavage
the
and
dystrophin
dystrophin
alone.
is important.
Right?
And it
And it
decreases
works
the
with
virus
patients.
titer
Yes,
and
yes.
myocarditis. And so there's this model here that demonstrates, I won't walk you through it again, but
Yeah.
the difference being the stability of the membrane, which limits the amount of virus that can leave and infect other cells. And this is the one that summarizes the clinical implications that I just reviewed. So at this point, we're convinced that the genes play an important role. And now how can that virus play in there? And I think that's an important new view. So we're just talking about these autosomal recessive cardiomyopathies that presented as acute myocarditis, pediatrics, and they had an increased incidence of either autosomal recessive, compound heterozygous, or homozygous variants that higher than would be expected in a normal population. Again, indicating that perhaps a genetic defect, including some cytoskeletal proteins, that could be involved in the pathogenesis of myocarditis. So I showed up at Intermountain Hospital, and one of the first patients that I saw was actually a 60-some-year-old man who was there with severe cardiomyopathy. He looked awful, in association with a skeletal myopathy. He was there with his surgeon friend, his cardiothoracic surgeon friend, and he wanted to make sure that we were going to take care of him right. And he had a sister that had died suddenly. He had a brother that had died suddenly, and he was there. And he looked terrible. He had been genetically tested, but had not found anything. I said, well, how long ago were you genetically? Well, it was about 5-6 years ago. So, well, we need to do that again. And everything was, his troponin was elevated, and his cardiac MRI was non-diagnostic, partly because he had a defibrillator placed, because of the family history. So genetic testing was done, revealed that he had a pathogenic variant in plakophilin 2, arrhythmogenic right ventricular cardiomyopathy, desmosomal protein, as you all probably know. And so, and there may have been significant variants in alpha-1 syntrophin and limb domain binding protein 3. This I think of as the Ju-Chen protein. It has a different name now than he named it, Cipher. But nevertheless, these are skeletal muscle and cardiac muscle proteins that are important. It may have explained that part of why he had skeletal muscle and cardiac muscle disease. Fortunately, we took care of him, gave him treatment that we normally would for heart failure patients. He got better, but we found also, we screened his family with cascade testing. He has a daughter that I now care for in her 30s with type 1 diabetes, actually, and ARVC with a variant in plakophilin. She recently had a defibrillator placed as well because of increasing arrhythmias. So why was the patient fine for so many years and then suddenly developed cardiac and skeletal muscle disease in his 60s? His father actually had the exact same variants in his 90s with occasional PVCs. Why does the daughter have recurrent myocarditis with elevations in her troponin? But sometimes she's fine. So the desmosomal proteins start to come into focus here as we start thinking about myocarditis and genetic predispositions. That was highlighted also by this paper here, which I think is a key paper, to be honest with you, to help us focus on why desmoplakin and maybe other arrhythmogenic cardiomyopathy proteins can cause cardiomyopathy. What they found in these patients with desmoplakin cardiomyopathy, 107 abnormal patients, 40% had evidence of myocarditis by cardiac MR. 52 of the patients demonstrated primary LV involvement. It's not just right ventricle. It's a cardiomyopathy problem, not just right ventricular cardiomyopathy. 4 acute patients underwent FDG PET, and all were positive for active inflammation. This is the PET scan, PET CT. Here you see the active inflammation in the left ventricle, and also here at the apex in these, and there's the cardiac MR showing the late gadolinium uptake in a pattern consistent with myocarditis. So let me tell you about two sisters in Utah. These are two people that were referred to me because they had myocarditis, recurrent myocarditis and pericarditis. So I said, I was talking to this one lady and I said, I think I know what you have. Her father had died suddenly in his 50s. Father died suddenly in his 50s. I think you have, I actually thought it was a desmoplakin variant for some reason, because of that paper, I think. Anyway, she ended up having desmoplakin variant. She has a sister that she referred to me that also has a desmoplakin variant. Sister A had intermittent periods of chest pain and troponin elevation, but usually she does pretty well. Sister B, who was the referral, had not had any significant symptoms, but she met criteria for arrhythmogenic right ventricular cardiomyopathy with the variant. Recently, Sister B called me and said she's having her first episode of chest pain. I said, you know, I might have just written it off, but I knew she had this genetic abnormality. I said, you need to go to the emergency department. And sure enough, her troponin was slightly elevated, but clearly elevated. The next day, her sister called me. Actually, the ED called me. She said, Sister A is here with a pretty severe episode of chest pain and a troponin of 1.8. It ended up that a few days earlier, they'd been together with each other. And I'm trying hard to figure out what's in common there, but I haven't been able to figure it out yet. You know, they weren't sick, etc. But it's interesting that something precipitated this onset of myocarditis with these two sisters that have desmoplakin mutation. What is that? We don't know. So do desmoproteins alone, abnormalities alone cause myocarditis? Is it the genetic defect that causes a skeletal muscle abnormality, a cardiac muscle abnormality that activates inflammation? You know, it depends on how you look at it, right? If you're a geneticist, you say, obviously, desmoplakin abnormality is the problem. Well, desmoplakin abnormality is a problem and is a key to the problem, but it is not the only thing, is my thinking. Something happens that precipitates that as we go along. So the different view of how we look at that same elephant. Or can there be an environmental trigger? So it's interesting that one of the, Neil Bowles is here now, I believe. And he was first author on this paper at Jeff Tobin's laboratory, where they looked at patients with arrhythmogenic cardiomyopathy. And this was in, let's see, 2002. And sure enough, there was a higher incidence of virus infection in patients with arrhythmogenic cardiomyopathy. Interesting. That was long before we knew that association between desmoplakin and myocarditis. Or does a combination of genes and environment affect the onset of myocarditis, which is what I think is the case. We're still learning more about that. Now, let's shift for just a minute about immune function. A lot of what I've showed you is about virus infecting the heart or abnormalities in cytoskeletal proteins affecting the heart. Is it possible that activation of the immune function alone can cause myocarditis? Well, you might say, of course, you know, autoimmune, but there are two examples now where it's very clear that only activation of the immune system causes myocarditis, and two models of that. One is, I'll go to the bottom one here, immune cell stimulation using checkpoint inhibitors, i.e. Javid Moslehi. This was his nice paper in the New England Journal of Medicine that demonstrated that initially. And this is a big problem with these great chemotherapeutic agents that activate the immune system, but definitely can cause myocarditis. Also, now with CAR T?
Not so much the full-blown like BT and stuff.
Yeah.
But they certainly get like, we've had a couple consults for like weird arrhythmias in patients shortly after CAR T therapy.
I believe it.
Yeah.
I believe it. And then now we're learning a lot about SARS-CoV-2 vaccination, right? And SARS-CoV-2 vaccination can be associated with myocarditis. These are direct immune stimulators that are causing myocarditis. So in this case, there's no virus. There may be a genetic predisposition. We haven't gotten that far yet, but they definitely activate the immune system. So again, important parts. This is the paper that Dr. Moslehi, who just moved to San Francisco. You probably all know him. One of the nicest people in cardiology, right? Everybody's a friend with him. And he's just moved to San Francisco to help build this program. but he started this field, published the first things on that. And it's a rare complication, 0.27 versus at 0.17. Myositis can also occur, etc. And it's especially prominent with the two agents, nivolumab plus ipilimumab. This is checkpoint inhibitors that do that. Now, this is a paper that we just published. It's one of several. It's not the first one, although we were one of the first ones to recognize it. And it's one of those situations, you try to get it in quickly, and JAMA says, oh, we're not sure you need to do this, and then they publish somebody else's the next week, right? So, we published this paper here showing a small but significantly greater incidence of inflammatory heart disease after vaccination for severe acute respiratory syndrome coronavirus 2. And this was done by looking at all adult patients over 18 at the Intermountain Healthcare Facility, any Intermountain Healthcare facility in Utah between December 15th, 2020 to June 15th, 2021 with a diagnosis of pericarditis or myocarditis. So that's when the vaccine started, about December 15th. We had 1.7 million patients that we were able to analyze, and in the electronic medical record. So analyzed means we look for the diagnosis, right? It's not like we looked at 1.7 million patients, but we pulled it out of the electronic medical record, which is where the power of this is. But we did have to analyze a lot of patients that had been diagnosed, but we did a retrospective analysis. 914,000 had received at least one dose of SARS-CoV-2, and there were 21 of these patients that had received SARS-CoV-2 probable or confirmed myocarditis or pericarditis or myocarditis. Interestingly, we found a lot that had some manifestation of pericarditis. 19 out of 21 were either pure pericarditis or myopericarditis. Only 2 had myocarditis alone. I think it's a little bit of a misnomer. People talk about myocarditis in these patients. I think most of them are myopericarditis, to be honest with you. Most of the papers don't look at the pericarditis phenotype. That gives us, and we looked at those who didn't get it in virus vaccines, and the relative risk was the absolute risk was 2.3 per 100,000 vaccinated patients. So if you have a patient wondering if they should get vaccinated, the risk of myocarditis is 2.3 per 100,000. That's very rare. And the risk of the disease itself causes much more disease than that. And the relative risk was 2.05 within 30 days. Importantly, though, none of the vaccine-related myocarditis, pericarditis patients died, with the exception of one who had a severe hematologic malignancy. And most of them were out of the hospital within a couple of days. It is not a severe disease. Let's talk for a minute about therapies for myocarditis and immunosuppression. This is the famous myocarditis treatment trial. Jay Mason here at the University of Utah was the lead author on this and organized it. And unfortunately, there was no difference between prednisone or other immunosuppressive agents. I think Imuran or others were included here. They couldn't find any difference in the cumulative mortality or the ventricular function in these patients. People say there are problems with this study. I would agree, but it's the only thing we have for generalized immunosuppression. And was using the diagnosis of myocarditis when I was a fellow, which was many years ago. 1987 to 2001 is when they were doing the study. But there's been renewed interest in this because of studies on cardiovascular disease. Canakinumab, an IL-1 beta inhibitor, was shown to have significant improvement in the primary endpoint of incidence of cardiac events in patients with atherosclerotic coronary artery disease. And that was kind of exciting, kind of proved the point that that activation of the immune system could play a role in atherosclerosis, but also has been stimulated more specific treatment strategies. There's a drug called anakinra, which also is an IL-1 inhibitor, that was shown to improve pericarditis, recurrent pericarditis, number of events with placebo. This is a very difficult drug to treat or to take. It has a lot of injection site reactions, and it has to be taken very frequently, and patients hate it. This drug here, published in the New England Journal of Medicine at the beginning of January 2021, in which we were able to participate in, showed that was studying rilonacept, which is an IL-1 alpha and beta inhibitor versus placebo, And the median time to the first pericarditis in these patients with recurrent pericarditis, they never reached it. Only 2 patients had recurrent pericarditis. All the rest of them, as compared with 74% of the others. And this graph is just profound. This is a great treatment. The only problem is $20,000 a month is the retail price on this monoclonal antibody that they're using. So it's pretty hard to give to patients. It does work. It does work. So now we know that it can be virus. It can be immune activation. These 2 models, we'll have to add T-CAR to this as well. Genetics can play a big role. We can get damage to the myocardium, and the amount of damage can affect the extent of myocarditis and the activation of the immune system. There can also be these direct immune, either autoimmune or these other stimulants that can activate the immune. All these can play a role in helping us to get there, to helping or in causing the myocarditis and the heart disease that can occur. Now along comes spring 2020. Suddenly COVID-19 dominated much of our thoughts and activities, didn't it? We didn't go anywhere. I was actually home. I was at home. That's not a picture of me. That would have been a long time ago. I had that much hair. But nevertheless, I was at home quarantining for no good reason. We now know, somebody in the lab had, or somebody in the clinic had COVID. So I thought, well, I'll quarantine myself at home. I'm sitting there with not a lot to do. And I'm just reviewing the literature related to COVID. And I'm thinking, I need to get into this. So we started studying. I jumped into the field, called some of my colleagues there at Intermountain, said, I'd like to get involved in this. It's interesting that because of the COVID pandemic, Intermountain mobilizes clinical trial resources like it's never happened before. Sam Brown has a big role in that, if any of you know him. This was by mid-2021. These are COVID trials that we had been involved in and enrolling at least 1,500 patients in COVID trials. The first one that we published, this is the first paper that I was honored to be for author in New England Journal of Medicine, was this one here, which showed we were testing monoclonal antibodies against COVID in hospitalized patients. And it ended up, it did not improve. This antibody works in outpatient, but it did not work on inpatients. Had no significant effect on hospitalized patients. And there are reasons for that. We know now because, active virus, it inhibited the virus, but when they come into the hospital, it's a lot more inflammation and less virus mediated. So what does the COVID-19 system do to what does COVID-19 do to the cardiovascular system? Well, we learned people who have cardiovascular disease are more prone to adverse events when infected. Patients hospitalized with COVID-19 can have cardiovascular complications that include elevations in troponin, pulmonary embolism, stroke, and other vascular thromboses. When patients have cardiovascular events in the hospital, they are more prone to severe disease and death. This is a paper published in JAMA Cardiology in March of 2020. It's amazing the amount of literature that's come out in 2 years over all this, isn't it? If you had an elevated troponin T with cardiovascular disease, this is the mortality. Without cardiovascular disease and elevated troponin T, this is the mortality, and so on. So any evidence of heart disease and evidence of previous heart disease contributed to that, the severity of their outcomes, as the risk of their outcomes. So that's important. So what are the potential mechanisms for cardiac injury in COVID-19? As it was shown that these patients had myocarditis, suddenly I got a lot of telephone calls from people saying, Kirk, have you seen COVID-19 causes, SARS-CoV-2 causes myocarditis? Tell me about myocarditis. This is a different kind of myocarditis. Some people have even said it's not myocarditis because there's not T-cell infiltration like there is with Coxsackivirus or adenoviruses. It's different. And it involves a number of different mechanisms that could be involved in cardiac injury. They can include microvessel dysfunction, coronary artery dysfunction, endothelial cell dysfunction, macrovascular endothelial cell dysfunction. It can include cytokine storm with lots of different cytokines, which could affect the heart. There's macrophage infiltration, much more than T-cell infiltration. When there is some virus in the heart, and there have been recorded cases of fulminant myocarditis, So all these different mechanisms probably play a role in the cardiac injury that occurs with SARS-CoV-2 infection. Now, is that myocarditis or is that not myocarditis? I think, myocarditis means inflammation of the heart. It doesn't mean T-cell infiltration of the heart. So I kind of side toward, cytokines. I'm okay with cytokines causing heart disease or heart injury. That is inflammation of the heart in my mind. So I think we should be a little bit more liberal in our definition, and maybe we should use the word inflammatory heart disease, or maybe if it's inflammatory, like some of these are, if it's caused by virus, maybe we should call it viral heart disease. But it's too late to change. I think everyone thinks myocarditis at this point in time. So, and that's partly what I'm addressing here. Many of you are probably familiar with this story that was published early, looking at 100 patients that had recovered from COVID-19 and found an extraordinarily high incidence of abnormal cardiac MR. 71. The interval was 71 days, and 67 of these recovered from home, and 33% required hospitalization. And these patients, very high incidence of abnormality. These are people who have been, this is their troponin, high sensitivity troponin levels in these patients. And it gave everybody an alarm. And that's when they closed down football practice, right? Is because they thought everyone would have this. And a total of 78 patients had abnormal cardiac MR findings out of the 100. 78% of people who had recovered from COVID. We're studying that now, aren't we, in many ways? So does SARS-CoV-2 infect the heart and cause myocarditis? Yes, but there is the factors associated with myocarditis cardiac inflammation, are different. And this is an autopsy series, 41. We had a good discussion here. We were writing a paper together with the university, with Robin and with others here. About myocarditis, and we thought, man, we wish we had tissue. Well, finally, there was some tissue that was obtained. 41 consecutive autopsies at Massachusetts General, people that died of COVID-19. There was cardiac infection in 30 out of 41. So there was infection in the heart in many of these cases that died. The number of cells were rare, however, and only 4 out of 41 cases met the classic criteria for myocarditis. And that's why people say they don't think it's really myocarditis, but there were a lot of macrophages and some lymphocytes present in the majority of the cells. So they say without myocarditis, I would want to say that is inflammation in the heart. So that is a degree of myocarditis. And here's some pictures that were from that. This is the immunostain for the SARS-CoV-2 that tended to be in the endothelial cells. I'm going to skip this one. This came out more recently and showed that there was an association between COVID-19 and myocarditis. And this showed 16 times higher incidence, with the risk though was very low, 0.146. And I'm going to skip these. This is having to do with the athletes. When they tested them, and ranked them, only 5 athletes actually had inflammatory heart disease based on cardiac MR, but not all of them got cardiac MR. And so, so there is some, but it's not very severe. So we usually let people play, and I won't go through. There's a broad bit of literature on who should play and who should not play after coronavirus infections. So there is a small but definite number of professional athletes with myocarditis post-COVID-19. In spite of the title prevalence of inflammatory heart disease among professional athletes, cardiac MR was only performed in 27 of the 789 athletes. Consequently, it's hard to compare to Putnam et al. There were no adverse events in 759 that had normal initial screening. So patients that die from SARS-CoV-2 infection, the virus is frequently detected, however sparse, tends not to be found in myocytes. There's evidence of inflammatory heart disease in many patients. A few have more typical myocarditis, but a higher number of macrophages is typical. So what have we learned from COVID-19 and heart disease? Viruses and inflammation can cause heart disease through many mechanisms. we're thinking in different ways now. We need to think about myocarditis and how it's caused in a different line of thought. Viral diseases can be successfully treated with immunomodulators and antivirals, antibodies. Immune mechanisms are likely to be important with viral infection, even if there's not a lot of cellular infiltration. And again, when we think about myocarditis, you actually have to ask yourself, what part of the elephant is that person examining? You may have been asking, why does he have a picture of Mount Kilimanjaro on many of his slides? It's because I think there are still mountains to climb. We're making progress and learning more about viral inflammatory heart disease in humans as well as mice, but there's still more to learn and more to understand. So we still have to climb Mount Kilimanjaro. Thank you very much.